Hello students, I am Dr. Santosh Upnekar. Now we are going to learn about the comparative embryology. In my previous lecture we already learned about the eggs and its types. Now in this unit first of paper second, it is a comparative embryology. The second subunit is dedicated to the structure and types of the sperms. So now we are going to learn detail about the structure and the types of the sperms. Now students, we are going to see the second subunit of our first unit, comparative embryology, and it is nothing but the structure and types of the sperms. Okay, first we see the structure of the sperms. Here I give you the one example of the sperm, and it is a human sperm. When we just see in this human sperm, it is haploid in nature, means the half number of the chromosome is present. Means we generally know that there are the 23 pairs of the chromosomes are present in a single cell, but here inside of this there is a 23 chromosomes are present. And this sperm, when we just see, having four parts. First is the head. You can see here the head, then the neck, then middle piece, and the tail region. When you just see inside of this head, a very prominent nucleus is present. On the top of this nucleus, his acrosome is present. This acrosome having a enzyme which lyses or the breakdown the wall of the egg during the fertilization process. Then inside of the nucleus there is a DNA is present, and just beneath of the nucleus you can observe here one tiny white body is present, and it is nothing but the proximal centriole. And then the another such type of the centriole is present, but it is present in this portion, means the neck portion. This neck portion, when you just see here the neck portion, it is a quite it is a depression we can observe just beneath of this head. And here actually the presence of the second centriole body, it is known as the distal centriole, and from this distal centriole the axial filament is formed, and this axial filament is nothing but the skeleton of this sperm. Okay, there are the 9 plus 2 microtubules are present, and they give the motility they give the structure of or the skeleton of this sperm. And then here in this middle piece, here the mitochondria are present, and they are arranged in a spiral fashion. So generally we know that the mitochondria is nothing but the powerhouse of the cell, and it gives the energy to this sperm for motile purpose. And this is a tail, so this is a nothing but the flagella. Okay, and we further discuss about the flagella, the uniflagella, biflagella, or the multiflagella, on the basis of the how many flagellas are present to the sperms. On that basis, we can divide it sperms. Okay, so the mitochondria give the energy to the this sperm, and last piece is nothing but the tail region. It is a tail, and due to this tail, it motile in a nature. Okay, so this is about the structure of the sperm, and this is the structure of the human sperm. Then here is also the same picture of the sperm, but here we can observe very detail, and you just see here this is a microtubule. This is a two microtubules are present at the center, and nine they are just at the periphery region of this. Okay, and these are the mitochondria. They arrange in a spiral like, and the you just see here the where is actually the cytoplasm. Okay, this is a nucleus, and this is a plasma membrane. Acrosome is present, and all these things are here, no doubt. But cytoplasm, it is a very small amount, and it is present at the tail portion. tail region. Means here, uh, in, just beneath of this plasma membrane, here the cytoplasm is present. Hence, the just above this microtubules. Okay, let's move to the next one. Next slide. And this is a slide is showing us the types of the sperms. When the previous slide we observed the human sperm, but uh, you can see here there are the number of the sperms are present on that. Uh, they having their different structure also. Okay, when we just to the its head portion, then the head portion is also different. We just look at this amphioxus sperm, then the domestic fowl, then this rat. Okay, compared with this human and this frog. Okay, so a number of the structure we can find out inside of this animal sperm, and also the flagella also. Okay, some sperms having a single flagella, some having the two and multi flagellated sperms also we can observe in animal body. So let us see the this type of the sperms. Okay, it varies from species to species, and when we just compare the size, then it is a very in Amphioxus the size of the sperm it is a 0.018 mm in a diameter, and in a toad it is 2.25 mm. Then we just see in the previous slide the head portion of this Amphioxus it is a spherical, and in also human it is a slightly oval or the spherical in shape. Okay, spheroidal in raw in a rod and lancet shape in amphibians, then spoon shape man and the other mammals. Means I just mentioned this spoon shape, oval shape also, nothing but the oval shape. Okay, and the hook shape head portion of the sperm we can easily observe inside of the mouse and the rat. Okay, then the sperms having a flagella for the movement purpose, or the for getting the movement, and so that this flagellated spermatozoa uh, or this flagellum we know it is as a tail. Okay, it is a flagellated or the biflagellated. We observe this biflagellated sperms in a toadfish. Okay, and in some cases there is a no flagella is present, or the flagellum is present, or the tail are present. Okay, so such type of the spermatozoa we can easily observe inside of the ascaris of the crab. Okay, then the uh, suppose there is a single flagellum is present, it is known as a uniflagellate, and suppose it having two flagella, then they are known as biflagellated sperms, and suppose more than two, they are known as multiflagellated sperms. Okay, now we can observe this multiflagellated sperm in a crustaceans also. Okay. In uniflagellated sperm, the head differ in shape according to the species. We already seen this sickle shape, and then the such type of the shape of the uh, head portion of the sperms in previous slide. Then let us move toward the next subunit, and it is nothing but the type of the cleavage. First, I just want to clear here that what is meaning of the cleavage? Cleavage is nothing but the cell division, which is going on inside of the fertilized egg. Okay, up to the formation of the fetus or the embryo. So zygote, when we just see here the fusion of the sperm nucleus and the egg nucleus, they share their chromatin material, and the zygote process is started. When the such type of the fertilized egg uh, it's inside of this fertilized egg, the cleavage is started. Cleavage means nothing but the cell division. Okay, this cell divided, redivided, and here is the formation of the blastula, then the after the gastrulation process, and then the actually the three germ layers also produce, and after the production of the three germ layers, the organs mm, formation process going on inside of this zygote. So the this is done by the process of the cleavage or the cell division. So let us see the in cleavages means what? Uh, During these cleavages, this zygote rapidly divide and redivide, but it is in a mitosis cell division process. Mitosis means the number of the chromosome is retained or maintained as it is in a daughter cell. Okay, such cells those are formed in mitotically in zygote or the fertilized egg. Here is a they are known as a blastomeres. They are known as a blastomeres. So remember one thing, that the blastomeres are nothing but the cells. Those are formed inside of the fertilized egg during the formation of the zygote. So in the cleavage, this is characterized by the rapid and successive mitotic division without any intervening period of the growth. Means what? Okay. Uh, The mitotic division is going on in this fertilized egg, no doubt, but the this large cell is divided and redivided into a number of the n number of the smaller cell. Here, there is no any type of growth is observed inside of this zygote. Only the number of the cell is for number of the cell is increased. Means there is only very large cell is there first, and then it converted into very small cells, means the blastomeres. Means total size is remain as it is, and the volume of the embryo remain as it is, but only the number of the cells are increased. This is most important or the essential thing. So the division of the cleavages is going on inside of the zygote. They are in a various planes. Okay, and they have different pattern, and this pattern is particular relationship to each other. And this cell typically tend to divide it in equal part. Means the daughter cells, they are equal in the size also. Means each and every new plane of the division tend to intersect the previous plane at the right angle only. This helps to maintain the spheroidal shape of the blastomeres. Okay, so during this cleavages, the spheroidal shape of the blastomere is retained or the maintained. Okay, the first cleavage division is often along the polar axis of the egg, and this cell is this is first meridional clip, and the second division is also the vertical and takes at the right angle to the first cleavage. Okay, and after these two cleavages, first or the second cleavages, the third uh, cleavage is occurred at the horizontal position or the equatorial position. Um, in this third division is horizontal, no doubt, but either at the slightly at the equator region or the slightly above the equator region. I just show you here. Okay, so here the first cleavage and the second cleavage at the right angle, and the third at the horizontal. But in this case, it is slightly above the equator region, and in other eggs we can observe this is at the exact at the equatorial region. Means the blastomere size of this blastomeres of these cells, they are same. Uh, Same. Okay, so the this this is totally depends on the quantity of yolk and the distribution of yolk inside of the egg. Previously, we divided the eggs on the basis of the quantity of the yolk and the distribution of yolk. So, okay, so the cleavage is whether it is going very fast or the slow, it depends on the how much quantity and the distribution of the yolk is present inside of yolk. Okay, generally we observe this division of the cleavage faster at the animal hemisphere and relatively slower at the vegetal hemisphere. Vegetal hemisphere nothing but there is a yolk is present. Okay, that's why it is known as the vegetal hemisphere, and the animal hemisphere is nothing but the where actually the nucleus is present. Okay, so, in some cases, whether the large amount of yolk is present, then the cell division may be incomplete. Okay. Thus, the cleavage occurs more rapidly at the active cytoplasm, means yolk-free or alethal or the microlethal egg. Okay, rather than the yolk-laden cytoplasm of the egg, mesolethal or macrolethal. Okay, and on that basis, this cleavage is classified into the holoplastic and the neuroplastic. Okay, so in we or the placental animal, where the nourishment occur or it is provided by the mother's body only, means this fertilized egg, it is resting at the fallopian tube and again rolled down and come inside of the uterine wall, and it plugged inside of the uterine wall, and the placenta, it is covered this entire fertilized egg, and within this placenta the all the nourishments and the oxygen, food supply, all these things occur through the placenta. So inside of this egg of the placental mammal, there is very small amount of the yolk is present or the yolk free also. So inside of this the holoplastic cleavages we can observe, means the very fast cleavages or the same size of the meroblast we can observe inside of this holoplastic or the inside of this placental mammal, means yolk free or the polycythal egg. Okay, and when I just see the holoplastic cleavage, again this holoplastic cleavage is divided into equal holoplastic and the unequal holoplastic, and it depends on the symmetry. Again we divided this into the on the basis of where actually the in which manner this cell divided or the blastomeres divided, and they are further divided into the bilateral, radial, spiral, and the rotational. We will see into next cleavage. So here the equal holoplastic cleavage is in a microlithical or the isolithical eggs, where this yolk is evenly distributed inside of this egg. So the in entire egg divided divides produce cells of roughly the same size, means equal, approximately equal in the size, means all the cells are the equal in the size, means cells, remember one thing, that cell known as the blastomeres. Okay, both the first and the second mitotic division the meridional and at the right angle to each other. Hmm? Then the plane of the third division is the horizontal and at the equator region. Okay, and as a result, 8 equal blastomeres are formed, which divided further to produce the approximate equal sized blastomeres. Especially we can observe in the amphioxus and the placental mammal in we also. Okay, so here in equal holoplastic cleavage, which depends on the type of the egg, means the amount of the yolk, uh, means amount of the yolk it is very less in this holoblastic cleavage or the equal holoblastic cleavage process. So distribution of the yolk is mostly important for this cleavage. So the blastomeres or the cells, those are formed inside of this after in this process of the cleavages. They are approximately equally sized. Then this is a picture. Okay, here the first and second and the third cleavage, and the equal size, approximately equal size of the blastomeres or the cells you can observe in this. Then the unequal holoplastic, it is just opposite to the equal holoplastic cleavage. Means in this mesolithical or the telolithical egg, means meso and the telolithical egg having the more amount of the yolk is present. That's why here the size of the blastomeres they call the micromeres, and the formation of this blastomeres, they are in a two types, the micromeres and the macromeres. So means the uh, at the animal bone where the cleavage is started, uh, these cells are size of the cells are smaller than the yolk laden blastomeres, means the macromeres. Okay, both the first and the second mitotic tubes are meridianal, no doubt, and the right angle to each other. But in the third division, it is slightly above the equator region. It is a horizontal, no doubt, but it is slightly above the equator region. As a result of that, the unequal size of the blastomeres you can observe. Then, so that there is a difference in between the macromeres and the micromeres on the basis of yolk. Okay, here we can observe the third division. Here we see it is just above the equator region. That's why the why, because here the yolk amount of the yolk is present, or the yolk is present just this or this position, so that the when you just compare the size of the lower or the these blastomeres, they are larger than the upper ones. So there are the four different types of the holoblastic cleavages, which depends on the cleavage forum. Bilateral, radial, spiral, and the rotational. Just see one by one. Bilateral, this is when cleavage actively is the same on the both sides, creating left and right walls. Means they are the very similar. Okay, means in the first and second cleavage. First bilateral holoblastic cleavage bisect zygote longitudinally into the left and right halves. The blastomeres of the successive division are arranged along a single plane occurring during first meridional cleavage, division into the two halves, which are mirror image of one another. That is nothing but the blastomeres are arranged in bilaterally symmetrical manner. We can observe this vertebrates and some molluscs and some echinoderms. Now, this is a picture showing then the radial cleavage. The second pattern is radial cleavage, which is when cleavage occurs radially around the central axis. At it occurs in an equal holoblastic division of the egg. In radial cleavage, the cell division are equal and symmetrical and occurs at the right angle to the polar axis of the egg. The blastula is divisible into the mirror image along axis drawn on any radius. Okay, we just observe in the C star, C arch, etc. Just to see, this is the animal bone, and this is vegetal bone. Vegetal bone means there is a yolk is present, and when you just to see this, this is the first cleavage, and then the second cleavage at the head plane, the third, and the animal. Okay, animal half and the vegetal half, then the mesomeres and the micromeres. The micromeres, we can observe such type of the division of the cleavage is going on, but at the radial. Okay, so this is the unequal radial holoplastic also we can observe here, and the spiral cleavages again. This is when the cleavage occurs in the spiral pattern around an axis only. Okay, the resultant cells are placed alternative to each other or in a spiral fashion. This type of the cleavage usually occur in the equal holoplastic type of the cleavage, although some undergo unequal holoplastic cleavages also. Okay, so we can observe here. This is a spire. Okay, you can observe such type of the division of the cell is going on inside of this cycle. Then the rotational cleavage. It is very interesting. In third pattern, actually third pattern is called the rotational cleavage, and this happens when there is a rotation of the cleavage plane. Here, the normally the first division is along meridional axis, giving rise to two daughter cells. Okay, but during second cleavage, one of the daughter cell divided meridionally, while the other equatorially. The rotational cleavage is found in the isolacical type of the mammalian egg. Okay, you can observe here such type of the rotational cleavages. And then the next one is a neuroplastic cleavages. In neuroplastic cleavage means the incomplete division due to the presence of the large amount of the yolk present in the macrolithical egg. You can easily observe in a tululithical and the centrolithical yolk. Yolk amount is very large. Okay, there are the two main types: discoidal cleavage and the superficial cleavage. Okay, discoidal and the superficial cleavage. You can observe this. Discoidal cleavage means in many animals have the discoidal egg which contain very little quantity of the cytoplasm as compared to the huge deposition of the yolk. In these cells, the cleavage is restricted to only at the cytoplasmic disc, means at the animal pole. Okay, hence the name itself is nothing but discoidal cleavage. Okay, in birds, fish, or the reptile, yolk is large amount of the yolk is present and very rich quantity of the cytoplasm is present, and it is shifted toward the animal pole where the nucleus is present, and at that position the cleavage is started. So it is just look like a disc. Okay, just see here in this picture we can observe. Okay, the cleavage is started right here at this animal pole only. Uh, so why? Because this is nothing but the this below portion is nothing but the yolk. So all the cleavages is started or occurred in this animal pole only. That's why it is just look like a disc. Okay, then this is actually the comparative discoidal, which is in the fish, reptile, and the birds. In telolithical bilateral cephalopods, mollusca. So we can observe this the bilateral. Bilateral means here the first cleavage, second cleavage, third, and all this process going on. And the central is the superficial. Most you can observe this into the insect. Okay, just see the superficial cleavages. Superficial cleavage is limited to a thin surface area of the cytoplasm that covers the entire egg. Okay, the center of the egg is filled with the yolk, which fails to cleavage. Thus, the cleavage does not take place in the cytoplasm, but only nucleus is divided through mitosis. Okay, so such type of the cleavage we observe in the cephalopods, mollusca. Here, the such type of the cleavage, and the next one is nothing but the superficial cleavage. Sorry, superficial cleavage. You just observe the superficial cleavage. This is superficial cleavage because very little amount of the nucleus is cytoplasm is present there, and the nucleus is shifted toward the animal pole, and entire this your egg is covered by the yolk only. Thank you.